Welcome to the EAU podcast.
Dear friends, dear colleagues, it's a pleasure for Dr. Contieri and I to be today at AUA, where we have the honor to have Professor Bivalacqua, who is today presenting a very expected trial. So thank you, Trinity, for being with us today.
My pleasure.
It's a fantastic times in bladder cancer. We see so many news in MIBC and in NMIBC with a lot of new drugs. And today you have presented one cohort with patients treated with cretostimogene. Can you tell us a bit the drug? Because most of the non-opinion leaders in bladder cancer don't know the drug yet.
Right.
And the rationale behind it and how it came to this trial.
Yeah. No problem. Happy to share with you our first results. So later on this afternoon at the SUO, which is the Society of Urologic Oncology, we're going to present Cohort CX, which is part of the CORE-008 trial. So the CORE-008 trial is a multi-cohort trial, which allows us to be able to study different disease states for non-muscle invasive bladder cancer. So we've already enrolled Cohort A and B, which is BCG naive and BCG exposed, treating them with cretostimogene. So what cretostimogene is an oncolytic adenovirus, which overexpresses GM-CSF in RB mutated tumors, which have essentially alterations in E2F. So this allows tumors to have a dual mechanism of action to prevent bladder cancer from coming back. So if it's an RB alterated, it destroys those tumors. But more importantly, and I personally think the real mechanism of action is that it activates cytokines. So GM-CSF goes up, you activate this influx of cytokines, which brings in T cells, macrophages, and you get a charged immune microenvironment, which I think ultimately leads to potentially that sort of prevention of the adaptive immune resistance with therapy.
So one of the characteristics is that it's not based only on alteration,
Correct.
but also on the implication of all the cytokines that you have.
Yeah, absolutely. So the adenovirus actually has the cassette, which allows us to insert GM-CSF, which then gets upregulated when it gets taken into the bladder mucosa. So when we place it into the bladder, you first have a detergent, which will release it, which allows there to be better penetration of the adenovirus into the bladder lining.
Okay, so you were talking about placing into the bladder. How does it work with this drug?
Yeah, so I'm personally more excited about Cohort CX than the other cohorts because it's actually a combination therapy. So what it allows us to do is it allows us to evaluate in two disease spaces, BCG-unresponsive and BCG-exposed, with 55 patients, randomized to cretostimogene alone or given with combined with gemcitabine. Two cohorts. One got gemcitabine and cretostimogene concurrently, so at the same time. So intravesical combination at the same time.
Okay.
Or sequential. So 2 weeks creto, 1 week gem, 2 weeks creto, 1 week gem.
With installation each
time. So induction, right? So induction once a week for 6 weeks. And the question we asked was, number one, is there a synergistic effect with gemcitabine? Potentially synergistic or additive, or is there really no effect? And would this combination not have any improvement in efficacy in BCG-unresponsive and BCG-exposed patients?
I think we see how the cohort works. What did you find? Yeah. What are the results?
So the findings are from an efficacy standpoint.
Yeah.
Now, the thing that I'd like to point out to everybody listening is that this is a 6-month median follow-up is 6 months.
Okay, so that's
super important.
Very early results.
Very early results.
Of this phase 2.
Correct. So we're not looking at 2-year CR rates, but what we found was, all the intention to treat, so all patients treated, both with concurrent as well as sequential, at 3 months, the response rate was over 90%. And at 6 months, it was 90%.
Nice.
So at any time, the complete response rate at any time, at any given time point, was 85%. So we are seeing a really pretty impressive, efficacy with this.
EFS was the primary endpoint.
Primary endpoint is so because this is not a registration trial. So let's make that point there, right? So it's a phase 2, 55 patients. So it's not registration, but because the FDA has stated that complete response at any time is sort of the primary endpoint that we're using for all of these trials, and the complete response rate at any time for these patients in this cohort thus far is 86%, but when you take out the patients, and this is sort of the important part for everybody to know, is that we lost 4 patients to tolerability. So when we had patients that were treated with the concurrent, same time, we had 4 patients dropped out because they weren't able to tolerate.
Because the tolerance is more difficult for patients when you have the same time combination therapy.
Absolutely. So what we learned is that probably not thinking about, for example, if the results at 12 and 24 months are still encouraging and still good, if we can think about a phase 3, we're probably going to need to do it with sequential, not concurrent, right? And what we're currently being considered in SWOG, so which is the Southwest Ecology Group, which is one of the cooperative groups here in the United States, there is a randomized control trial looking at patients with BCG-unresponsive CIS plus or minus papillary disease, where they get randomized to creto with gem or gemdosi.
Okay.
Interesting. So that trial has not been activated, but it's being for consideration in the cooperative group setting.
And I think it's very important because gemdosi in that setting is not that much, I would say, assessed in a randomized clinical trials.
Yep.
So it's highly used in US. We are having more and more patients treated as well in Europe, but it's still difficult to get and to administrate gemdosi in Europe because it's not as good as in US in terms of dissemination.
Yeah, but it's been the uptake in Europe's pretty. It's moving forward.
It's moving forward. What a question. So you will think that if we have to give first creto and then BCG, for example, or gem, how do you see it? Because the patient needs to come more often to the clinic, right?
Yeah. So because this is like induction, so weekly for 6 weeks, they're coming. They're coming once a week for 6 weeks, just like BCG or any type of intravesical chemo. So the workflow is the same. Okay. The only difference is cretostimogene, because it's a adenoviral gene transfer, you do have an extra step of placing the detergent in the bladder prior, right? So it's a DDM. It's placed in the bladder, dwells in the bladder, and then you drain it, and then you place the creto. So if you're giving it concurrently, that's a long session for that patient, and that's probably why it's not tolerated as well.
Maybe another question. So the BCG-unresponsive and exposed field is moving forward super fast, and many drugs try to find a place for avoiding cystectomy. And we all agree that we try gemdosi, whatever, re-challenge BCG, single chemotherapy sometimes for some people. And many drugs are trying to take this market. And how do you see that creto has a role in it? Do you fall to the DOR will be something that will be important for the drug?
Yeah, so I think right now, if you look in the United States, and listen, it's being approved. For example, other of the novel therapeutics that have been approved by the FDA in the United States are now being approved in Europe. So I would love to be able to see that creto if got approved here, because remember we're talking about an unapproved medication, right?
Yeah.
I mean, or intravesical therapy. My understanding is it's currently at the FDA. I suspect it will get approved because the Bond 3 trial, which was the pivotal trial, which was the phase 3, was it's actually in press at Lancet Oncology right now, showed that at 24 months, creto, intravesical instillation, which is weekly for 6 weeks, it has the ability to do reinduction if you want with maintenance, which is the SWOG protocol. The disease-free survival or CR rate at 2 years was 45%. Okay, so that's pretty impressive. That's better than any of the others. So if you can bring that 45% and get it to be 60% by combining it with another intravesical agent, I mean, which is gemcitabine. Anywhere in the world, you can use gemcitabine, right? I mean, it's being utilized everywhere. So I'm most excited about this. Now, as far as your question about BCG-exposed and BCG-unresponsive, Listen, BCG exposed is a disease state that we have developed in the last 2 years, right?
Yeah,
And especially in the U.S. A lot of drug companies or pharmaceutical, big pharma are trying to move into that space. However, you got to do a large randomized controlled trial in that space to show that it is effective against, for example, BCG, because those are the patients that we're going to be giving more BCG to. So I think in order for any type of intravesical therapeutic to be approved in that disease space, they need to undergo a large randomized controlled trial and probably randomized to either BCG or potentially chemo.
Yeah. Yeah. Thank you very much, Trinity, for your time. I think you have pursued us that there is still a lot of awaiting new strategies that will be able to change the field, especially in BCG and then BCG naive and BCG unresponsive patients in the coming years. So thank you very much for your time. And we hope to see you soon.
Absolutely. Thank you so much.
Thank you.
Thank you for listening to this episode of EAU Podcasts. For more updates on bladder cancer, please visit our educational platform, UROONCO Bladder Cancer. To keep up with the latest EAU podcasts and stay informed on the urological advancements, be sure to subscribe the EAU podcast channel on your favorite podcast app. Until next time, keep learning and stay inspired.